Pyruvate decarboxylation. The citric acid cycle can only process 2 carbon molecules, and pyruvate is a 3-carbon molecule. PD is an enzyme-catalyzed reaction that takes the 2 pyruvate molecules and converts them to 2 acetyl coenzyme A molecules. These are 2 carbon molecules. Energy is stored during PD by converting 2 NAD+ to 2 NADH, and the extra pyruvate carbons are expelled as CO2, or carbon dioxide. Now we'll take a look at the citric acid, or Krebs cycle. After glycolysis and PD, 2 molecules of acetyl coenzyme A are further processed in the citric acid cycle. This takes place in the mitochondrial matrix, the innermost space within the mitochondria. The diagram on the left shows one cycle, which is due to one acetyl coenzyme A molecule. To account for one glucose molecule, 2 cycles are needed. So this is one turn of the cycle due to one acetyl coenzyme A. Note the production of 1 ATP, 3 NADH, and 1 FADH2. But one glucose molecule yields 2 acetyl coenzyme A molecules. Therefore, 2 turns of the cycle yield 2 ATP, 6 NADH, and 2 FADH2. Click here for a video on the citric acid cycle. Remember that the citric acid cycle is sometimes called the Krebs cycle. The cycle breaks down 1 acetyl coenzyme A for each turn, generating 1 ATP, 3 NADH, 2 CO2, and 1 FADH2 per acetyl coenzyme A. Since 2 molecules were created, or 2 acetyl coenzyme A molecules were created from each glucose, the citric acid cycle produces 2 ATP, 6 NADH, 4 CO2, and 2 FADH2 for each glucose molecule.